Well, welcome everybody. My name's Rebecca Wadham. I work at the University of Virginia, and I'm really pleased to be here to give the concluding talk in the respiratory pathogens webinars. It's been a really great series. I've really enjoyed some of the talks, and I hope I can do justice today to follow on with what everybody else has presented. Some of the things I wanted to go over today is this will be the bacterial part of the respiratory pathogens. And as you can imagine, there are a number of bacteria that are included. So it'll be, more focused on on some specific bacteria. But I wanted to give you a brief overview of the PATRIC resource and all the bioinformatic resource centers in general. And then I thought, because this is the last one, it might be a good idea to give you an overview of all the respiratory pathogens. I started looking into this, when we give these different ones, we tend to focus on different organisms, but I wanted to look more broadly at what are causing respiratory diseases across the globe and who the principal players are, and if there are differences between different regions across the globe. Then I was going to go in and look at their two important respiratory pathogens that are bacteria, Staph aureus and Streptococcus pneumoniae. I have an example of an analysis of taking a group of Staph aureus genomes that I got in a publication, and I'm going to step you through very quickly of how you could do this in the PATRIC resource. And then the next one is there's an excellent review paper of the virulence factors related to pneumonia in Streptococcus pneumoniae. And this will be a little bit different, but it's something that people generally have a huge problem with. And I myself have a problem with it. When you read something in a publication and it describes a gene and it doesn't give you the sequence, and it doesn't give you a GenBank or a RefSeq locus tag, how do you find that thing? So I'm going to show you some tricks on how you can do that and how PATRIC can help you. And then, of course, I'm open to any questions you might have. So for those of you who don't know anything about PATRIC, you may be asking, what is PATRIC and why is it spelled incorrectly? Because doesn't PATRIC usually end in a K? PATRIC is the Pathosystems Resource Integration Center. It is one of the BRCs, that's what we initialized them down to, that NIAID has funded since 2004. And by NIAID, I mean the National Institutes of Allergy and Infectious Diseases. What it is a data resource designed to support analysis and comparison of bacterial pathogens, but we have all bacterial genomes, all the public ones. So it's not just for pathogens. Because when we first started this, it was how do you know something's pathogenic what the differences are unless you compare them to non-pathogenic bacteria. So PATRIC has all the data, and we go to GenBank regularly and bring it in. Now, when the BRC started in 2004, there were 8 resources in the first round of funding. And in the second round of funding, it went down to 4, the bacterial, the viral, and actually it was 5 because the viral had the flu database and the virus pathogen database, the influenza research database, which is IRD, and the virus pathogen database and analysis resource, which is Viper. But in my mind, they're both virus, so I combined them to just think the viral ones were another one. Eukaryotic pathogens like malaria, giardia, that's in UPathDB, and vectors of disease like the mosquitoes, ticks, lice, all those things that are transmitting agents to you through the bite of those insects. So there were 4 in the second round. Now in the third round, they've combined down to 2. PATRIC is now combined with virus and IRD, and the eukaryotic pathogens, UPathDB, and vector-based are now combined. The new bacterial viral BRC, which we call BV, bacterial viral BRC, is currently being built. And you're getting a special sneak preview of what it's going to look like. We expect to announce it later this year, but will be the landing page, and it'll give you easy access to the resources that you may not know or you do know, and hopefully, we hope you love them. So you'll be able, or like them, or okay, just use them. That's what we really want. So this will be coming later this year. The teams that bring you Bvbrc, we have 3 teams. One from the University of Chicago and Argonne National Lab. So that's combined into one major group. J. Craig Venter Institute is the second group, and University of Virginia, the group I belong to, is the third. So these are all the people you can praise or blame for what's happening with the Bvbrcs, but hopefully it's a little more praise than blame. So I want to just walk you through some of the main features that PATRIC has, and why we think it's special and why we think you should use it. Well, the first thing is it's this database. It's a whomping big database. It's a large solar database that houses all the data. And we used to use a different database, but solar makes it really fast, so you can search for things really quickly. So what data do we have in PATRIC? Well, it's mainly genome-centric, and we also have the things that are annotated on those genomes, which are genes, proteins, intergenic regions, things like that. We also have the metadata associated with those genomes, and we have a bunch of other omic data. We have protein families, transcriptomic data sets, metabolic data. There's an intense amount of information in PATRIC. Now, as I told you, we have all the data. We bring it in from GenBank. We have regular updates from GenBank and different sequencing centers. And we also go to the short read archive and bring in genomes too that are of interest to us that we think are going to improve your experience in PATRIC. And for an example of this is we've made a concentrated effort over the past 4 years to go in and look at publications that are describing antimicrobial resistance and large studies that are looking at that. Generally, those people put that data in the sequence read archive, not in GenBank. So we read the papers, and we've been trying to train machine learning models to recognize areas of antimicrobial resistance. So we grab that data from the SRA, we assemble and annotate it and bring it into you in PATRIC so that you can see it. And this is the only place outside of the publication that this data exists. Of course, it exists in the short read archive, but you can't really examine the genomes, but we bring them into PATRIC. Currently, we have more than 442,000 assembled genomes. Now, this includes bacteria, archaea, and we also have bacteriophages that we've brought in, and some host data as well, the principal host data sets, so that like you can do RNA-seq if you're looking at a response of a host cell to a particular bacterium, and that you have those fast Q reads, you can analyze that data in PATRIC. Now, one thing that I think is our superpower is we provide a uniform annotation that enables our comparative analyses. So we bring the data in, and then we annotate it using RAST, which some of you may have heard about. So why do we do this? We do this, PATRIC, you can consider it the Rosetta Stone. We bring in the data from GenBank, but we don't lose that data. We keep all of their identifiers of genomes. We keep their gene identifiers so that you can make those comparisons. But in order for you to be able to do large-scale comparative analyses everything has to be similar. You have to be speaking the same language. So we translate everything into PATRIC so that you can call on our protein families and look across everything and look for presence and absence of those, rather than having to do BLAST and multiple sequence alignments to see if you have the same thing. So that's why we do that. Another thing that we have in PATRIC is we've created this comparative analysis environment. So if you're a biologist, which I am, I'm not a programmer, we provide ways for you to see that data and understand it. For example, here we have the Compare Region Viewer that I'll show you later today that's showing you gene neighborhoods and how they're conserved across different organisms. We have whole genome alignments, which uses MAUVE, which some of you know about, and this is the PATRIC design for that. And I really think it looks pretty. It's so crisp and clear. I'm not going to go over that today, but yeah, I encourage you to go look at these things. We also have multiple sequence alignments for you to use. These are just some of the viewers that we have. We also provide you with the ability to get all the data. The bioinformaticians routinely in the early phases of PATRIC, they found it easier to get the data here than any place else. So they would just come in routinely, download all the data, and go away and have their way with it. So they were mainly doing this through the FTP site, which you see over here. But once the University of Virginia and the University of Chicago were more matched together in the second phase, University of Chicago is strongly computer-oriented, and they weren't going to mess around with that. So we developed a command line interface that you can use too. Now that we have more and more bioinformaticians, we provide you with easy access of you to come in through the command line, grab the data, and also launch a number of the services and use our pipelines to analyze the data. So this is the thing I think that makes PATRIC truly unique, and this is the part of it that I'm most proud of, and I think that sets us apart from everybody else, is we provide you with a bunch of ways to analyze your data. Generally, we try to pick things that are computationally difficult, that will take more than you can do on your individual computer. You have a private workspace. You submit the data through your private workspace. We handle the compute, and then we give you back this structured output. And I think what shows that, indicates that we're increasing in use is in 2009. I mean, I don't even want to talk about 2004 because we only had 8 genomes at that time. But in 2009, you can see that the public genomes were picking up, and this goes into 2018. When we started these services that you can see over here in 2015, suddenly we started seeing an increased rise in the private genomes that people were bringing in to analyze to PATRIC. This is several years old now, right? Now they're neck and neck. So the amount of private data is up there with the public data. Oh dear. There we go. So this just shows you how those services are being used. In 2015, and the services that were available then are indicated by this dark blue box. We started with Annotation and Assembly, Proteome Comparison, and RNA-seq. So, we were proud of what we had in 2015. In 2016, we could see things rising. And that year, we brought in the variation service, which is SNPs and genomic regions that have flipped and things like that. And then it increased more in 2017 when we brought in phylogenetic trees and metagenomic binning. 2018, another increase, we brought in this service, which is the Comprehensive Genome Analysis service. Used to, when you would do an assembly, you had to download the contigs and upload them again and do the annotation. And then you would have to grab genomes and do a phylogenetic tree with that. Comprehensive genome analysis, we realized that people were tired of all the clicking. And so that combines those two things. So we brought that in, and you could see it really started to increase. In 2019, we brought in taxonomic classification, metagenomic remapping, and FASTQ utilities. 2020, we brought in whole genome alignment. So you could see this huge increase in number. And in 2021, just the first 3 months, we're already here. So we're expecting to have a bumper crop of jobs submitted this year. Notice that in every single year, the primary workhorse is annotation. So often we have people who've assembled their genome somewhere else. They bring the contigs in and annotate them, not only so that they can get the RAST annotations and the things that come with that, but so they can experience the comprehensive genome analysis environment that we have in PATRIC. So, bioinformatics is hard. It's especially hard for biologists like myself. And so we provide you with different ways through this help section on the top tab to get help. There are user guides. We have tutorials. We go through common tasks. For those of you who are bioinformaticians or computer savvy, you will probably want to look at the command line interface tutorial and just and use that. We provide webinars, instructional videos, and workshops, and we also have an online class that's free through Coursera that I'll talk to you about in a little bit. So whenever you have a question, you can just click on Contact Us. It opens up this page, and you can type in a question, and we should respond to you within a couple days. Not on weekends so much, though. So that's enough about PATRIC. I'm assuming that you all came here more to hear about the respiratory pathogens. So I was going to give you a sort of summary over the principal players in these important diseases. So some data from the World Health Organization tells us that the lower respiratory tract infections are among the top 3 causes of death and disability in both children and adults across the globe. They think that nearly 4 million deaths occur annually because of this, and it is the leading cause of death of children under 5 years old. Moreover, when those kids get infected and they have if they survive it, and they've had a serious illness, it predisposes them for chronic respiratory diseases later in life. So it's sort of disabling them too. Just as with influenza as an example, influenza kills between 250 and 500,000 people annually and costs between $71 and $167 billion US annually. So we're talking about a serious cause of morbidity and mortality on a global scale. The disability adjusted life years, what are those? That's the sum of years of potential life lost due to premature mortality and the years of productive life due to disability. This was a study from the Lancet that was comparing the leading causes, looking at changes between 1990 over a 20-year span and 2019. You can see that the respiratory infections were number 2 in 1990. In 2019, they're number 4, but they're still in the top 10, and they're still a major source of misery across the world. So what are respiratory infections? Actually, I'm sure that everybody knows this, but I thought I'd step through it. Generally, there are things that happen in your nose, your sinuses, your pharynx, your bronchi, and in your lungs. Oops, I went back. So let's talk a little bit about your nose and the common cold. Everybody gets that, right? Everybody gets colds. Mostly colds are viral, with rhinoviruses accounting for most of them, followed by influenza virus and then coronavirus. I'm taking data here from Mandel. I can't remember the year of this particular book, but it's the 6th edition. This is all the data you're seeing is pre-COVID. So, that I'm sure has changed a lot of data, but we're just going back to 2019 in our minds before we had to deal with the coronavirus epidemic. Sinusitis, when you get those intense headaches because your sinuses are blocked, whereas things happening in the nose are predominantly viral. When you get into the sinuses, you have a mix of the major players being bacterial or viral. The major players in sinusitis, it would be Streptococcus pneumoniae or Haemophilus influenzae, but there's strong viral component too with rhinoviruses. So we've got rhinoviruses in the nose and we've got them in the sinuses. Moving down the respiratory tract, I don't know if you ever saw that old film, Fantastic Voyage, where all the scientists get in this little spaceship and they're shrunk down and then they travel through the body. Well, we're sort of doing that through the respiratory tract. When you hit the pharynx, when you have sore throat, you start coughing stuff up. That's often, that can be mixed bacterial or viral, but it's predominantly bacterial, well, mostly. You have this time Streptococcus pyogenes. And then, of course, here come the rhinoviruses again. They're traveling down the respiratory tract with us. But it's interesting to me how the different players with the bacteria are not the same. I would think, you'd be swallowing this stuff, and strep pneumonia would also appear in the pharynx, but this time it's strep pyogenes. We travel down, looks like we're passing the trachea and going immediately after the bronchus branches into the bronchi. You have different viruses here, which are the predominant players in causing disease. And this time, while rhinovirus is present, for the first time, we're seeing respiratory syncytial virus and the parainfluenza viruses. So all of a sudden, viruses come back into play. And then I don't know about you, but when I think about respiratory infections, I generally think about pneumonia. I had pneumonia when I was a kid, so that's probably why I would think of that. Now, there are all different kinds of pneumonia, and there are different pathogens that cause it. So depending on the type of pneumonia, it's going to be a little bit different. But the major players, the bacterial, are strep pneumoniae, Staph aureus, and Haemophilus influenzae. From the viral side, it's different on if you're an adult or a child. Adults generally are getting the influenza viruses and adenoviruses. Kids are getting hit with the respiratory syncytial virus, the parainfluenza viruses, and also influenza A. So let's talk a little bit about the types of pneumonia. Well, you can have community acquired, which is when, all of these things have very strict definitions. So if you come into the hospital and you already have pneumonia, that's community acquired. If you come into the hospital, you're admitted, and then within 48 hours, you develop pneumonia. They think that that's community acquired. From the bacterial side here, because I just got data from bacteria, this is the Bacterial Respiratory Pathogens webinar after all, so sorry if I'm going to focus more on this. From the bacterial side, the major causes of community-acquired pneumonia are strep pneumoniae, Klebsiella, Haemophilus. Now, if you've been in the hospital for more than 48 hours and you were never intubated, that's hospital-acquired pneumonia, and that's a couple different pathogens are causing that. Staph aureus is entering for the first time. We all hear about staph and MRSA, and you see those scary things on TV and about MRSA, and also Pseudomonas aeruginosa. Now, if you've had intubation, it's the same thing, but it's now called healthcare-associated pneumonia, and that's Staph aureus and Pseudomonas. And then if you've been on a ventilator, that's called ventilator-associated pneumonia. So I wanted to take a look at the prevalence of these things across the globe, and this was a really excellent article by Torres et al. In Nature Reviews looking at community-acquired pneumonia. Now, when you look at let me go back up. When you look at the United States and Europe, the majority of the cases, they couldn't detect a pathogen, although the person did have pneumonia. But when they could detect it, in the United States, it was more viral, and in Europe, it was more bacterial. In India and China, there wasn't, there was some no pathogen detected, but they're primarily bacterial. So what's causing what? Let's first look at the viral cases, just to give virus its due here, and also because these authors covered it. In the United States, which has more viral than bacterial community-acquired pneumonia, there are those rhinoviruses again. But when you look at Europe, it's influenza that are causing most of the pneumonia cases. And in India, it's influenza. And in China, apparently, they don't have as much viral pneumonia as we do, although I find that surprising because they get flu too. Everybody gets flu, right? Influenza, sorry. When we look at the bacterial side of it, in the United States, 11% of the cases are bacterial, and it's strep pneumoniae. Europe, strep pneumoniae. India, strep pneumoniae. China, Mycoplasma pneumoniae. So it's just interesting to me how, depending upon where you are, the major players can be a bit different. Then I was looking for other studies across the different kinds of pneumonia, and there wasn't anything quite like that Torres review in Nature Reviews. But this was a pretty good study from Eni et al. From 2014, where they compiled a lot of data from different places to look at the summaries over a certain amount of time. And you can see that in different places, we're now concentrating on bacteria again, the principal cause of disease is different. So for hospital-acquired pneumonia in Japan, it's predominantly Staph aureus. In Brazil, it's the same. But in Asia, across Asia, well, it's split between Staph aureus and Pseudomonas aeruginosa. And in Thailand, so this was just a different study than the Asia one, because Thailand is, of course, in Asia, it's predominantly Acinetobacter. So that's the end of my discussion on the respiratory pathogens. Any questions right now? Okay, then let's get into examples of an analysis in PATRIC. So I wanted to do two different things. I wanted to recreate a study I saw of Staph aureus and also a different study on Streptococcus pneumoniae virulence factors. This was an excellent study by Steiner et al. In Australia. So they went in and collected genomes between 1992 and 2009 of Staph aureus genomes that were methicillin resistant or associated with methicillin resistance. This was causing a fatal necrotizing pneumonia, and it was killing a lot of young people. Now, what I really liked about this study is they provided their data in a bio project over in GenBank. So let's talk a little about what they did. They did assembly, they did a Snip analysis, they did a phylogenetic tree, and they did a comparative genomic study. So I wanted to show you how you could do that in PATRIC. So I want to get out. Can you guys all hear me? You sort of disappeared off my screen, and I want to be sure you're all still there. Yes?
Yeah, we can hear you.
Okay. All right. The little thing went down on me. Oh, while I'm here, this is going to be like a whirlwind tour of PATRIC. So, first of all, let me point out that it would help all of us out. Our sponsor, NIAID, is conducting this survey of the bioinformatic resource centers. So from the PATRIC website, if you would click on this and take the survey, it's very fast, and we would really appreciate your helping us out here. But you can click on these things and step through things. I have a new online course on Coursera that goes through, it's totally free, and it goes through a lot of the stuff. I'm going to be doing this very quickly today. So the course is here. You can enroll for free. I go over assembly, annotation, comparative genomics, tree building. I'll give you a whirlwind tour today, but I would encourage you to do this course if you want more of the details. And I'll be launching soon Bacterial Bioinformatics Phase 2 that will cover taxonomic classification, metagenomic binning, FASTQ utilities. Some of these things I'll be showing you today, and also RNA-seq. So anyhow, let's get back to Steiner's paper. Steiner's paper had the bio projects. I got so excited when something's been deposited in a resource, then I know I can get the exact date. I don't have to search for names. I don't have to struggle. So, oops, sorry about that. So the bio project was 232112. So there we go. All right. So it looks good. It was Staph aureus. It was Melbourne. That's Australia. The registration date looks good. And it's SRA. SRA means it's reads, not contigs. So are we stuck? Can we go no further here? Actually, we can go for a lot further. So let's click on this link. And this shows us all the genomes in their resource, not in their resource, in this bio project that are united in the bio project. But it's the sequencing, it's just the reads. So let's click on one and see what this looks like. So it was Illumina HiSeq. Here's all the stuff about it. Now, what we used to have to do in PATRIC is go in and download this data and then upload it into PATRIC. And these genomes are large, or these read sets are large, and sometimes that would take a long time, and it was very frustrating. But now you can just take this number, Control C, and let's go to PATRIC and see what we can do with it. One of the first things I always like to do when someone gives me data and asks me to analyze it, I work with a lot of lab biologists. They always assure me that this is a clean sample. They have streaked it so well. There's no contamination in it, and quite often there is contamination. So I always like to use the taxonomic classification service to check for that. So I click on that, and this uses Kraken 2, which is an algorithm that was designed by the Salzburg group at the University of Maryland. One of the things we like to do at PATRIC and also at Viper is we don't want to reinvent the wheel. If there are pipelines out there that the community, that the biologists really like, why don't we just bring them in and make it easy for you to do here? So that's what we've done with Kraken. You can use read files, or you could just upload assembled contigs. But these are read files. Now, if I had a paired read library, I could go in to my workspace. You have to be registered to do this. And I recognize that I'm going to be blowing through this stuff, and I apologize to you guys for that, but there are the tutorials and stuff that you can look at. So I could click here on this little folder icon, and then I could upload that data to the computer. If I've already uploaded it and I want to run it again, I could click the down button and see the ones that I've uploaded recently. Or if I know the name of it, if I didn't do it recently, I could start typing and it would give me auto-suggesters. So you can do paired reads, you can do single reads, you can do both and bring them over. But we have an accession number from the SRA, and see how easy this is? If you never had to do this before, you won't appreciate how great it is to just be able to do it this way. So I have the reads here, and I move them over. You have to move it over into the selected libraries. The only algorithm we have right now is Kraken 2. It's against all genomes. You can save the classified sequences or the unclassified sequences, those reads that are mapping to not this genome, but the k-mers associated with this genome. So don't make the mistake of thinking it's against an entire genome. It's just looking for unique k-mers that they have associated with this genome. You need an output folder to put it in. You can click here and create a new one, or choose one that you already have named before. Or if you've named something, which I have, I could just put it here so it's showing me all the different times I use this. Click on that, and then the output name. And I always like to put a date on it because I run these things again and again, so I know which one to look at. Today is 01 June 2021. For those of you who are international, often you should try to use the standard characters. If you use the ñ for Spanish or whatever it is in Portuguese, the irregular characters will throw this stuff off. So you notice when I named it, it suddenly became blue and I could submit the job. And so I submit it and it says it's submitted. Now this is going to be and it's already been queued. And in a little bit, I'll be able to see it on the jobs page. It generally has a delay of like 5 minutes before it does that. But this is like one of those old Julia Child cooking shows where she mixes the cake and puts it in the oven and then turns around and brings out another cake. Actually, she never baked anything, but you get the gist, right? So there's a number of ways to see your jobs. So I'm going to click here on my jobs icon here. And that takes me to my jobs page. And so I want to see a recent taxonomic classification job that I did. Note that there's nothing in this vertical green bar until I highlight it. And then I have two choices. I can view the job, or if it failed, I can report it, which we encourage you to do so that we can figure, it could have been that something happened at Argonne Lab and there was a system failure that caused jobs to temporarily fail, and we can restart those for you. Or you may have actual questions. So you do that, you'd click report issue, and then you'd say, I've had people say a lot of things like, this is so important to me, I have to get this done for my PhD I'm defending tomorrow, help. So hopefully you wouldn't be defending tomorrow if and depending on this one job, but then you could submit that. But we want to view the job. So I highlight it, click view, and this is the structured output that I'm telling you about, that I referred to earlier, that you get with any job you submit to PATRIC, any of the service jobs. So it took 1 hour and 5 minutes to run. There's a lot of things in here. Refer to the tutorials, but you can click on this. I wanted to show you this guy, the HTML report. And then you notice a number of things happen here that you can do with this. But let's just click on the view icon. And this is showing me the percent coverage of reads that map to the k-mers for that specific, in this specific read set. So you can see that 96% of them map to Staphylococcus, 90 to Staph aureus, and then as you get down to the strain names, it happens to hit this guy. Now this guy, they referred to this JKD6159. Steiner et al. In their publication talked, and they included this genome in all their analyses. So let's just take a quick peek at it. So I click on that, and it's going to take me over to it. And it says that there are 2 genomes here. So I can go here and click here. Oh, and I can see that one is a plasmid, and this is a complete genome. And I can see more information on that. And it was actually sequenced at the University of Melbourne in Australia too. Anyhow, let's go back to this. I wanted to show you one last thing because, you know, tables are great, but we also have this interactive chart from Kronos that shows you how many of the reads are mapping and where they're mapping. So I feel really good now that this genome is, or the reads in this genome are pure. There are no contaminants in it. So what did they do next? Well, they never did taxonomic classification, or of course they did do it because they wanted to find out where it was. One of the first things they did was they took each of these read sets and aligned them to this genome. So let's see this genome again. Over here, I can click on the genome landing page, and I can see that genome. And while it loads, I want to show you a service that you can do to align reads to a particular genome. So I open the services, and I go here to FASTQ utilities. Click on that. I would have to give it a output folder. There. I'd have to give it a name, and I would just call it that guy. And while I'm here, we'll go into the pipeline in a minute. I want to put it here for the accession, and move it over. Notice that I can't submit a job yet. Now, under the pipelines, I can do 1 thing, 2 things, or 3 things. I can trim the reads. If I know I have adapters or something on the reads, I can cut them off here. I can get a quality score for that and see how good the reads are and if there's anything I need to worry about. Or I can do an alignment against a different genome. So I would click on that, and I have to move it here. And once I move it here, notice that this box becomes active. It wants me to define a target genome. Well, the target genome we want is the same one that they used, which is this guy. And the easiest way to get that is to grab the Genome ID, which is a unique identifier that's really going to help the easiest way to name something. Because often there are currently 8 Mycobacterium tuberculosis H37 are the genomes in the PATRIC system because people keep submitting them and different labs with that name. This is specific to this genome. And then it's blue. I can look at it and I can check the quality of that job. So let me show you a different way that you can see a job. I don't need to submit this again because I've submitted these things a number of times, but this is just to show you guys how you can do it. I can go, but to find the submitted job, I can go into my workspaces and I would click on my home directory. And I've had this workspace since about 2009, and when I first started using it, and it also continued with me being very messy. So this is sort of like when someone comes into your house and they accidentally open your closet door when they're looking for the bathrooms or something, and all of your things come falling out onto the floor that you stuffed in so your house would look clean. That's what you're seeing in my workspace. I would encourage you to be a neater person than I, because I've had this workspace for a really long time and it's really disorganized and chock full of junk. Well, useful stuff, but so I united everything into this folder and FastQ utilities, and let's just look on any of these jobs that I did. If there's a little checkered flag, it means like with the race cars, this job completed successfully. So I click on that, and there are a couple things I wanted to point out to you. I can look here at this HTML and view that, and it's going to tell me how many of my reads mapped to this particular genome. And it gives you a bunch of other stuff, but I just wanted to show you that predominantly first, because people often say, how do I know how many of my reads map to the genome? Well, there's the percentage right there. So I can click on this too, and I wanted to point out, right now we don't have a way to an easy access to the genome browser from this. So you would need to download these files, but then I could download the BAM and the BAI file. And I want to show you something really cool that you can do with this. This is that reference genome that they used. And I can go into File, and I can say Open, and select Files. And I'd already done this before. So I'm going to open this, and this tells me that it's opened correctly. And I click the Open, and look, these are the read files that map to the genome. If you scroll down, there's regions that don't have, in this particular SRR number, there's a big region here that doesn't, that they have no, that the reads don't map to. So these are the RefSeq locus tags, and these are the PATRIC locus tags. Look, we have a transposase. So this looks like an area of horizontal transfer. And you can look over and see what those proteins are. Uh-oh, there's a penicillin binding protein. And there's a methicillin resistance protein. So the reference genome has those, but this particular genome seems to be missing them. I also wanted to point out that you can click on an individual read, bonk, and get all the information on that. So Steiner et al. Did an alignment. We did an alignment. The next thing they did was an assembly. So let's do an assembly. So let's go in, let's go back over here so we can keep this open. You could do an assembly uniquely or an annotation, or you could do them both at the same time and do the comprehensive genome analysis service. So, I've forgotten who that read file was. So let's grab him back, just so you can see that. Now I can start with read files or contigs for this particular one. I just move that over, and this is going to start to look repetitive to you, but each service has something a little bit different to offer. Now we're just doing the thing from the sequence read archive, right? But you could submit paired reads, single reads, or both together to do a hybrid assembly. So let's talk a little bit about the strategies you could use. If you know something about this, like let's say you just have Illumina, you could assemble that with unicycler or spades. If you know you only had you were sequencing a plasmid, you could use plasmid spades. If it's a metagenomic sample, you could use metastabades. If you had reads from single cell droplets, you could use MDA. Now, if you had long reads, it would be Canoe. Or if you did a hybrid assembly with long and short reads, you could either do Unicycler, which handles them both at once, or Canoe, where you could use the Illumina reads for the polishing. Notice that you can choose if I were to use Canoe for this, it would fail because these are Illumina reads, but I'm just giving an example. You can adjust the contig length. You can say, I only want to see things that are bigger than this. I only want to see things with this much coverage. Now we get down to the crucial part. All of this is crucial, but to me, this is most crucial. When you do an annotation in PATRIC, if you can get to the genus level, and remember, we already did taxonomic classification, so it tells you what the genus level is. Why do I want that? Because if you can get to the genus level with every genome that comes into PATRIC, we assign protein families that are Kamer-based. All genomes get the cross-genus families, which we call PG fams. If you can get to the genus level, you'll also get the PL fams, which are more fine-tuned to a specific genus, and you can do a more fine-grained analysis. So we knew that this was Staphylococcus. Oh, look, it's helping me out. Staphylococcus, I don't think I have to use aureus. I can put my identifier in. I can put in a folder, and I'm ready to submit. But I don't want to actually submit right now because I've done this a number of times. So let's go look at what a job looked like. Oh, and look, my taxonomic classification job already completed. So here's a recent comprehensive genome analysis I did on one of these. So I can click on that and view it. And mainly I want to show you how easy it is to do all of this stuff. So this is all the details for the successful assembly. This is all the details for the successful annotation. But what I really remember, you can go to the webinars, to the bacterial bioinformatics course, or any of the tutorials to give you deep down help with this. But in this whirlwind tour, I want to show you this full genome report. So I click view on that. And first, the most important thing, which it tells you straight up front if this is good or if you should stop and go back to the lab. It says it's good quality. Often we didn't have this before, and it would take me until I was doing the comprehensive analysis and actually looking at the proteins to look at it and say, oh my gosh, this genome's a mess. But this tells me right up front, and it tells you right up front that it's good. It gives you details on the assembly. It gives you details on the annotation. It tells where your thing, remember I just put in Staphylococcus, but it did an analysis of it, and it says it's actually Staph aureus. It's showing me what the proteins are like. And then it shows you this graph. So this is the genome sequence itself. These are genes on the forward strand. These are genes on the reverse strand. These are the ribosomal RNAs. These are genes with antimicrobial resistance, and these are identified virulence factors. Well, wait, you may be saying, where did that come from? How did you know that? Well, this is another thing that you're getting with the annotation in PATRIC. We blast every genome against specific databases that we have for antimicrobial resistance and susceptibility genes, and also for virulence factors. So it's showing you those as well. And there are other ones which we'll show you in a second that you get that aren't in here. And this shows you GC content and GC skew, which are indications of where you might have lateral transfer. Now you'll notice the break here. This is one contig. This is the second contig, etc. So you notice that these are different colors. Here's something else you get with an annotation. a subsystem analysis, because each of those genes are tagged with where we think they function in metabolism, protein processing, like these guys, all on the forward strand here, the red, stress response. You notice they're together. So you get a subsystem analysis of those. I talked earlier about the antimicrobial genes. These are the databases that we blast against. The Comprehensive Antibiotic Resistance Database out MacArthur's group in Canada, NARDO from GenBank, and our own special ones that we've been getting from machine learning and things like that. We also blast against the drug target database. And so this genome had 34 hits to that one, and this one to TTD, and also the transporter database. And then for virulence factors, the virulence factor database out of China, which is like the gold standard in Victors. It also, not only that, I feel like, but wait, one more. We give a prediction of what we think is going on with this genome, that it's resistant to these antibiotics and that it's susceptible to these. And then it shows you the genes that we think are important in virulence. And then it does a quick tree, really quick, only 5 proteins, 5 protein families. This particular service uses the cross-genus families. So please don't put this in your publication. Use the tree pipeline that I'll show you later. But it's showing you where your genome is in the great scheme of things. And more importantly, it's showing you the references. The way I design these things is I want you to be able to take this information, a lot of it, and drop it into your final publication. What's also, everybody's going to want to know what annotation system you used. So here it is. This is it. So you don't have to look for it or try to figure it out. How are those protein families identified? Here's the publication. What about these subsystems? I use that. What do I reference? Here it is. It's all here for you. I wanted to make it easy for you to write, because when you write these papers and you reference these people, I hope you will also reference us, and that helps both of us. We want to ease your path to publication. Let's go back one more thing to this. You notice these that you'll see that looks like a library of little books leaning over? I can click on that and it'll open the tutorial that will describe everything that I just described and step you through it with those garish, bold letters that'll make your eyes ache after a while. But it also has the references down at the bottom as well. Okay? So, Steiner et al. Did an alignment. They did an assembly. Now, the other thing they did was a SNP analysis. So to do a SNP analysis, you would go into the variation analysis. And, we've stepped through this a number of times, how easy it is to do that. So what I really want to do is point out we have different aligners that you can use. So you can compare, generally when I run these things, whatever I'm running here, I do it with BWA-MEM, BWA-MEM strict, and Bowtie2 is the aligner. And because I guess I have obsessive compulsive disorder, as the SNP color, I use Freebase, SAM tool. So I do every possible one. You notice I didn't say last, that's for long reads, and these are short reads. And then I'd have the same target genome and where to put it. So let's see what that looks like. And this can be really cool. So I really like the variation service. I haven't used it that much lately. I'm going into my workspace because I'm trying to get you used to thinking about different ways, they say that all roads lead to Rome. Maybe all pathways lead to your workspace. So there's the variation analysis. And the little checkered flag says we ran it all. When I submit that job, I could submit all those read files to it. but I'm hoping I'm just going to show you one here. So I'm going to click on this checkered flag, and it shows how long it took to run that. Oh, look, I ran them all here. Yikes. So it's got the BAM and BAI files that I could also look at on the genome browser, but I want to find that HTML file. So sorry, I'm going to have to scroll down for that little guy. I think he's going to be at the bottom. This is why it took 6 hours to run, because it's a monster. Now this one will tell me what all the translations are, but as this is the whirlwind tour of PATRIC, I want to just show you quickly what this HTML file looks like. So let's view that. All right. So it's starting with the samples, okay? And then it's saying for this particular sample, when I compare it to the reference genome, which is on this contig, and at this position, the reference genome has a C, this one has a T, and then it gives you the coverage of that from the read files, and it's a non-synonymous substitution. So when I look at the codon, it goes from a GCA to an ACA. Well, does that really affect the amino acid? It certainly does. Within the amino acid at position 40, it goes from a G to an A, which translates to an alanine to a threonine at position 14 in the amino acid. This is, the position in the gene. So this is the unique identifier for that gene, as it's called in PATRIC. This is the RefSeq locus tag. This is what it does, or what it's called. It tells you the upstream region, the downstream region, and it tells you what's happening here. Is it missense? Is it intergenic? It is frame shift, and it also ranks you as to what potential impact it has. So occasionally you'll see some with a high impact. This guy had a frame shift mutation within this protein. How cool is that? And you can download it. What I often do is I download this whole file because we provide it as a download file too. Here, let me show you. Well, down at the bottom here. I download it. I'm a biologist, right? So what do? I download it into Excel. And then I grab all that data, I sort it, I get all the high impact ones, and then I can create in a publication let me show you that real quick because it is totally cool, this guy. From here, I can go into the circular viewer for it, and I can upload a custom track of those genes that have SNPs. I mean, I gotta admit, I just love being able to do that. That's so cool. Okay, so Steiner et al., they did the alignment, they did the assembly, they did a SNP service, they also did a tree. So let's go. They say that only God can make a tree. Well, all of us can make a phylogenetic tree in PATRIC. So we go over to the phylogenetic tree service. Now, I'm not going to show you how to create a genome group right now. I'll do that in a few minutes. But rather, the first, if I wanted to add this genome in here, I would take that unique identifier and put it up here. And let's say you had 100 genomes, and you can imagine how tedious it would be to search up that identifier and add them one at a time. But you can also add a genome group. You can create those things together, and I'll show you how to do that in a few minutes. And click on that and add that to it. And then once again, output folder, output name. But another thing is the number of genes you want to use. 10 genes is relatively fast, 20 slower, 50 slower, 100 slower, 500, 1,000 can take days. But sometimes you want to use that, as I'll show you in a minute. Now, these original authors did SNP tree, which is about as fine-grained as you can get with this. What this particular pipeline in PATRIC, it's called the Codon trees pipeline, and what it does is first of all, it takes this group of genomes and it looks for the PG fams, the cross genus families, and it just brings this number up randomly. And from those, and it's looking for what we call perfect genomes, which are all the genomes have only one copy of the protein in this protein family. So it's grabbing all the perfect, the quote unquote perfect families. From those, it gets the nucleotide sequence and the protein sequence, and it concatenates an alignment of those. So it's actually a pretty powerful tree to do that. Now, let's say you have a genome that you really need to work with, but it's a cruddy genome, and you notice that it's got a lot of duplications. And so most of its protein families have duplications in them. You can adjust that to allow for that, or it's missing a lot of stuff. You can adjust this to allow for it. So let's look and see what these tree jobs look like. So I'm going to go back to my workspace, go home. I mean, you guys will be able to navigate my workspace yourselves by the end of this. Go here into the phylogenetic trees. So you noticed I did 100, 500, and 1,000. And here are my completed jobs. Let's first look at the 100. So I click on that, and here's all the stuff that comes in with it. And you can refer to the tutorials to figure out what all those things are. But I want to click on this report and view it. And here, oh, look, we have a tree. And oh, look, here it says you had 56 genomes and 56 of them had data. You wanted 100. We found 100. And this is what I like the best, right? How many amino acids did we use? How many nucleotides did we use? If you have a good tree and you put this, I review a lot of papers, and these are the kind of things I look for. If you put just even audaciously put this in the paper, no reviewer is going to question that, because that's strong evidence of the strength of your tree. 100 genes, these many amino acids aligned, these many nucleotides aligned. And it also shows you what more information on this, what the genomes were used and stuff. But look at this tree. You're so excited about it, and then you see, oh no, look at these support values. They're terrible. 0 means if I ran this tree another 100 times, it would have 0 chance of coming up with it again. This is terrible. I can't publish this. What am I going to do? Well, what it's telling you is that these things are so similar that 100 genes wasn't enough to distinguish between them. So let's go back here and let's look at what it looks like when we have 1,000. So let's click on that. Let's click here and let's view this. It says wait time is usually less than 10 seconds. It's stealing my da-da-da-da. Try reloading it. Okay. Oh, there it is. Ah, now I feel better. 90, 99, 100. Ah, and this one that doesn't have a value indicates to me that it's midpoint rooted. So this is a tree I could be proud of. And look at the support that was used for this one. So the amino acids and the nucleotides. So maybe some would quibble that this isn't as powerful as a SNP tree, but when you bring in these things and you have those support values, nobody's going to complain about that. Okay. One of the last things these guys did was showing you the pan genome and the core genome. Guess what? We have a tool for that in PATRIC too. So let's look at that. The tool we would use for that is called the Protein Family Sorter. So I can click on that, and the genome group, which I'm going to show you how to create in a little bit, I would do Australian, that was what I called it, Australian study, and I want to do PL FAMs, but you could do PG FAMs, but these are all Staph aureus. So you can see, like if you want to compare Bartonella to Brucella, I'd use the PG FAMs, but within Staph, I'm going to use the PL FAMs. So it loaded pretty quickly. This is the table. It's showing me the protein family identifier, how many proteins are in across this group, how many genomes have this many proteins, what it's called, min, max length, the mean, and the standard deviation. Now it says across this group there are 2,768 protein families. That's your pan genome. If I click this button, which is saying, oh, I want to see all the protein families that are shared across all of them, 2,439. That's your core genome. If I want to see ones that are unique to any particular family, which we might not see any of, I would click absent and all. This guy, he doesn't have any unique. This guy, he doesn't have any unique, but that's one way you could drill in. These are all so close. Now, another thing you can look at, we're going to go into more detail on this later, but these are those perfect families. So you could use this tool before you create a tree, before you wait 5 days to create a tree and figure out that it wasn't going to work. You would click here for perfect families and filter it. And then you would see that 2,642 are those perfect families, one protein per genome. So let's reset this. I wanted to show you one other thing that you can do here. So we have also a heat map view of this. So if I stretch this out, here are my genomes, here are the protein families. If it's black, it means 0. This genome didn't have this protein family. If it's yellow, it means this one had one copy of the protein in that protein family. This orange color, it means 2 copies. This tangerine color, it means 3 or more. So let me show you another cool thing that you can do. To try to see patterns in your data, you can anchor it on a specific genome, or you could cluster all the data together. All right, so it's completed. And here it's showing all, well, this doesn't, oh, there we go. There, that data is clustered. So it's showing you regions that are unique to some and missing in others. And there was one here that looked really interesting to me because it's got some antimicrobial resistance genes in here. So how do I see those? I want to dig into that. I want to see what the genes are here. You notice how I'm drawing on this and I'm able to capture that data? So I draw a little box over it, and then it gives me some choices of what I can do. I can say, I want to see that data. So I click on that, and then you can see the data, and I can click on a particular, these are all the genes in that protein family. And this one, like the penicillin binding protein, let's look at this. And that was, I was looking at this particular gene. Here's the protein in red. And you notice these things around it? Ooh, look at these regions of, this is methicillin resistance. This is erythromycin resistance, methicillin resistance. If you want to see how broadly shared this region is, you can click on this tab for Compare Region Viewer. And it's been a little slow today, but let's see. And so it's taking the gene neighborhood and looking across all the reference and representative regions in PATRIC and saying how conserved is this neighborhood across the whole of PATRIC. And you can see this is the gene I came in on, and this is the blast. We also blast, showing you the blast results against that gene. So that's the big box without the arrowhead. These that are colored the same way are all the similar protein families. So you can see that this guy is conserved across a number of genomes. These are the reference and representative genomes, which is a collection of about 8,000 genomes. You could change it to all public genomes, but I'm not going to do that because it takes forever to do that. Okay, back to the slides for just a minute. Okay. So what did Steiner do that we were able to replicate? Well, first of all, we did a purity check using the taxonomic classification service. We were able to look at alignment using FASTQ utilities. We were able to assemble and annotate the data using the comprehensive genome analysis. We were able to look at SNPs using the variation service. We were able to generate a tree using the Codon trees service. We were able to look at the pan, core, and accessory genome using the protein family sorter. And then I showed you how you could look across gene neighborhoods using the compare region viewer. Now, I hope you'll hang on with me for like another 5 minutes because I wanted to quickly show you things that are often difficult to do, but some quick ways to do that in PATRIC. So I wanted to show you how you can find data in PATRIC, go from a publication. So I started with this publication from Nature. This is an awesome publication, actually, if you like virulence factors. These guys did a great job in this review. And so they defined, let me just show you, the genes of the different that are important in upper airway colonization, competition in the upper airway, and respiratory tract and infection data. So this is just a great thing. But look, they don't have unique identifiers. Capsule, how am I supposed to find that? That's even if I looked for capsule at GenBank, that'd be a number of things. And CHOP-P, often it's so frustrating for me when people come up, well, it's not. The fact that they don't provide a sequence, sometimes it's really hard to find these things. So I wanted to show you what I did, because like I'm a biologist, right? I go in, I grab that data, and like a dog that gets a bone or whatever, I have to translate everything into Excel where Excel speaks my language. So I grabbed all that data and I did that. And what I wanted to find were the protein families that were used in this. So let's just choose one of these, like this HYL. Okay, I'm going to go back to PATRIC. I know you guys are like, PATRIC, I'm so tired of hearing this, but we've still got a little bit more. We'll be okay. So I could put the gene in there and just search for that using this little icon to see what I get. Oh dear, 4,607. Remember, we're talking strep pneumoniae now. This paper was on strep pneumoniae, not staph. So I could go here and filter in and try to find among all of these results exactly what it is, or I could try to refine my search, this way. Oh, Control V, oh, brother. HYL, the search button is now at the top of the box here. Streptococcus pneumoniae. Look here. All right, we have 8. Only 8, huh? Across all of those? Well, let's look at them. So I click on that, and what? There are only 4 here. It said 8. Well, what it's counting is both your PATRIC ID and your RefSeq ID. And it's giving me this protein family. If I click on this, I have more information about the gene that I can see, including its size and everything else. But how do I know that's the one they were referring to? Because I don't see that this is linked to a publication. What do I do? Well, generally, I go to a trusted resource for protein information, and I go to UniProt, and I would put in HYL, and then Streptococcus pneumoniae. Whoops. All right, and I don't know if you use UniProt. I could also do this at GenBank, right? But UniProt gives a lot more information, so sometimes I like to look at that. So look, 1067, this was 1067. Maybe, do I think that's it? That could be it. That could be it, but let's click on it and let's see what we can see about this. Okay, look at all the information that's in UniProt, right? It shows you where it's located, gives you all this information about it. It tells you what it was called previously, but what I really want is this, which is the sequence for it. Once I have that, if I have the sequence or if I had a GenBank ID, I could find it in PATRIC. So this was the old study. So I'm going to show you a different one here, and BLAST, the biologist's friend lot, right? Everybody loves BLAST. All biologists love BLAST. So I can insert the sequence there. I want the protein. And I can look at these particular subsets of data within my own genome, within a genome group, or within a taxon. And this would be Streptococcus pneumoniae. It's going to help me because I'm a terrible speller. All right. And I want it to be kind of specific. I want good hits. So let's see. Fingers crossed that this is our guy. There are a lot of Streptococcus pneumoniae genomes in PATRIC, so sometimes it takes a while to get there. And like if you were to do this at GenBank, you could go away from, it would inform you when it was done. Right now, we just have to wait until it's done. So we have to be patient here while we're waiting, because I don't want you, everybody likes to see this, and I often try to arrange the little bubble there, so the little spinning globe and spin within a spin. But rather than waste your time doing, well, not waste your time, to mesmerize you with that, I want to show you quickly how to create a genome group. So I could go into organisms, Streptococcus. These are for the major pathogen groups. Go into the taxonomy. And you can all watch this. It'll be available. So I know I'm blowing through things, but you can watch it in very agonizingly slow detail later. And then I would go to the taxonomy, and then let me go to genomes here, because there are like 25,745 genomes. Click on those. And how do I choose them? I could click on this filter, and let's just get all the complete genomes. Boop. And then I can create a genome group. And I'll create a new group called Complete Strep Pneumo. Add. And it tells me that's been completed. So now we're going back to the BLAST. Really good BLAST hits here. And if I want to see what it is, I can see the alignment looks good. But it doesn't show me there's the protein family right there. 1044, which was this guy, 1044. So we could go over and look more at this particular guy, or we could do it from here. Look from here at this particular protein. So this is the protein, the feature page, and there were just a few things I wanted to show you with this that I really like. I could see how many genomes in PATRIC have identical genes at the nucleotide level to this. So I can click on that. And so there are 94. How many have identical proteins? Click on that. 357. And one thing you can do here is, let's just to show you a quick thing. I could click up to 200 here, and I could generate a multiple sequence alignment. Nucleotide or amino acid. I just want the amino acids because it's very pretty. And I want you to see how pretty it is. It gives you a gene tree, and it shows you the sequence, and across the top, it's summarizing it. So, of course, they're going to be identical because I chose identical, right? In the local families, just in Streptococcus, how many are in this? So it's a lot bigger, right? And if I choose just another subset, I could launch the MSA, the amino acids. And then you can see getting a little bit more noise here, but it's just so pretty. And you can change the colors and stuff if you like. Anyhow, you can, there's a lot of fun things to do. Oops, where are we at? There we go. And then I can see the global family across all the genera in PATRIC, 58,760 protein families. I mean, proteins belong to this family. One other thing that I quickly wanted to show you, sorry, is we have links. If this was a representative or reference genome, you could link directly to String and Stitch, which are awesome resources. So I would suggest you try those out. But every gene should link to the conserved domain database, which I like a lot. That's through GenBank. And it shows you what domains are on that particular protein. Now, I want to ask for your indulgence to allow me to do one more thing. So let me go back here, and I want to grab these protein families, because often one of your questions is, here are all these virulence genes. In my particular group, who has those? So I could go back to the protein family sorter, the genome group I just created, and because those are PL FAMs, I need the PL FAMs and search for that. All right. Then using this filter box, I can put those identifiers in and filter on that. Okay. And then the easiest way to see that is to click on the heat map. And then, although the names, they're the protein family identifiers. So you can see that some of them are very broadly shared and others are not so. So this is a really fast way for any of your genes of interest. You can look at them across a group of genomes and see who has it and who doesn't. And so with that, I wanted to, what did we learn now? I showed you how to use the global search. We used PATRIC BLAST. We looked at all the different types of protein families, how many, the identical, the local, the global. You got to see links to other sites. We generated a bunch of multiple sequence alignments, and we got to use the protein family sorter to be able to show us which genomes in my group of interest had particular genes that I'm interested in. And that's the end of this marathon. Once again, remember to go to the tutorials. Remember, if you want to take that bacterial bioinformatics class. But I'm happy to entertain any questions if you all haven't just fallen asleep or run screaming from the room because you just can't take another second. So it's open for questions if you guys are still there.
Thank you, Rebecca. That was really comprehensive.
It went on a little long. Sorry about that.
No, not at all. Yeah, I mean, feel free to unmute yourself and ask your question in person, or if you'd like Rebecca to go over something, or you have a question about your project in particular.
You can also submit questions here to go to contact us. Oops, whoops, not that one. Sorry. Here, go to provide feedback. Sorry. And if there's not, that's fine too. I'm sure we could all do with some coffee, go for a walk, go for a run,
So
run
there was
away.
actually a question about how should they enroll in the class on Coursera. So maybe you could just
Okay,
show where the
I'll
link is.
I'll show that. So I go here and over here
So
you
that's
can
at
it
patrickbrc.org.
right the third dot over, or you could go into Coursera and just search for it up here. But whoops, where was I? From up here. Oops, homepage. Sorry, guys. To the third link over, click on this, and this will give you a hyperlink to register to the course for free.
Okay. So there was another question that says, if we have sequenced the genome with a plasmid, how do we isolate the assembled plasmids? I guess how can you tell the difference between the plasmid genome and the chromosomal genome?
Okay, well, one of the ways, if I know what the plasmid is, or I know of a close thing, a different tool that I didn't show you today that's described in the course, there's a couple ways you can do it. If the plasmid's good and contained, you'll be able to see it in the assembly, and it'll be on a single contig in the assembly that you can probably recognize by the genes that are on it. So if I was at, let's see, what has plasma? Well, I'm just going to go to this guy because this guy's in two genomes and it's easy to see. I mean, it's not two genomes, two contigs. So from the genome browser, I can see either contig, right? So or you could go into sequences and see either of the two contigs. So you can blast that against the known plasmids. Or another thing you can do, something I didn't show today, which is the proteome comparison. And let me just show you what those jobs look like when they complete, because they're gorgeous. Let me see if I hope I have a good one. Oh dear. Well, I'm just going to try this one and see what it looks like. Generally, I have my favorite ones here, but it gives you this circular view of the thing where I could submit a plasmid sequence to it, and then it would show how strong the homology is here. And I can also download it. The strength of the blast hits are here. So this is a region of low homology. But if I was using the plasmid and compared it to this, I could show it to you in detail. So we have part of the bacterial bioinformatics class goes over this because this is an awesome tool, especially the Excel sheet that comes with it. It's just, it's beautiful. But you could also look at the tutorials. So that would be the way I could do it. Also, actually, thinking about it, I could do the genome alignment tool if I knew what the plasmid was, and I could select, some people use this tool and they put in like 20 genomes and it produces this mess. But I would use it with much fewer genomes, and you could see that. Let me see if I have a good one that I could show you. Genome alignment. Some of the ones with Brucella are quite pretty if I have um, let's see what this looks like. And then just want to view it. So, oh, isn't that pretty? Just love that one. So that can, I mean, if you knew what the plasmid was, and let's say I used the plasmid for that, then you would know exactly where it was, which of those contigs in your genome were that. Also, there are specific genes you can look for that identify a plasmid, but sorry, then it escapes me right now what exactly those are all.
So I think there's mobility, mob typing, and incompatibility typing.
Right. So
Right?
you can look for those particular genes, and they are generally at the start.
And this is my question, does Patrick have plasmid typing tools or not yet?
Not yet, but
Okay.
we have plasmid assembly up. Jamie Overbeek was working on that. I don't know if she's still working on it. Thanks everybody for coming. I know this was a lot. Remember that we have the user guides and the tutorials, and we also have some webinars. I think you'll like the bacterial bioinformatics class better though, because that breaks it down into like 10-minute chunks as opposed to when I first started doing the webinars, they were sometimes lasting, 30 minutes. And what you guys have just sat through, and it's hard to sit through that. But I think those things will help you. And so feel free to reach out to us with the provide feedback at any time. I'm here to help. And I appreciate your coming. And I congratulate Anna arranged this whole webinar series. And I think it's been fascinating and a great series of videos. And thank you so much for putting it together and inviting me to speak. Thanks a lot. Have a great week and a great summer.